This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1013, recorded on June 2nd, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 89 Fahrenheit, partly cloudy, and that is in Celsius. Oh, rats. I had it all pulled up, and then it went away. 32 Celsius, light breeze.
Yeah, it's 32 here and sunny. Yeah, very good. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Guess what? It's 89 here and sunny.
Very good.
Wow, 3 for 3.
Yeah.
And also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it is 91 Fahrenheit, which is 33 Celsius.
Wow.
And a few clouds, but we've got line of thunderstorms that might be coming in shortly.
And from Montreal, Canada, Angela Mingarelli.
Okay, so it's the coldest here, but it's actually 87.9, I think, which is 31 degrees, which is very hot for us. So we're all around the same.
We're all kind of in the same boat here.
Yeah.
This is amazing.
In the same boat. Well, we haven't done a TWiV for a couple of weeks, this kind of TWiV. And
You know, my notes say the last time I was around was 1007. Vincent, you've been jetting around, right?
Yeah, I've been, the last 3 weeks, I was in Switzerland, then I was in Maryland, and then last week, and then this week I went back. So last week there was a polio conference on Wednesday. Kostya Chumakov, who was a FDA guy, is the guy who retired last year, and Amy replaced him. She and George Belov gave a symposium in his honor last Wednesday, and they had all these old polio people back. It was just amazing.
Where was this?
It was at a hotel in Bethesda.
Okay.
It's actually the Pooks Hill Road, where you've gone for study sections, probably. It's such a funny name. And I mean, these people will probably never be in the same meeting ever again. You know, like Ann Palmenberg and Ellie Ehrenfeld, Jeff Almond, Vadim Agol come in. There were about 30 people there, Jim Hogle.
I'm trying to think of, there was a guy at CDC.
Mark Palanch.
Yeah. Was he there?
He zoomed in.
Okay.
Kara Burns came in person. And so I hired Chris Condian to record the whole thing.
Cool.
And we're going to publish it as videos on the Microbe TV channel. And then what else did I do? I did something else last week, but I can't remember what it was. Then I went back.
Well, you've got a whole bunch more coming up, because I'm not going to be on TWiV again until mid-July sometime. So.
Yeah, we actually have no more of these Zoom TWiVs in June. This is the only one. This week I went to. I went back I gave a talk at the Cosmos Club in D.C. It's a science club, and it was fun. You know, dinner. It's a dinner thing, but pictures on the wall of all people who were members of the club, and then members who won Pulitzer Prizes, and then members who won Nobel Prizes. It's pretty cool to see who's a member. I mean, you know, every famous person you can think of, right? We had Ruth Bader Ginsburg, Everett Koop, Walter Winchell. Everybody's there. It's really cool. And then I did a TWiV with Kostya yesterday. He has an interesting history, so we'll release that. So yeah, next week I'm going back. Rich and I are going to U.S. What is it?
USAMRIID.
USAMRIID. And we're gonna do a TWiV
Did you mention coming to Quebec, to Canada? You went to Quebec City recently,
I forgot.
right?
That's right. That's what I did last week. So I went from the symposium to Quebec, yeah. And I was at the last day of an aquatic virus meeting, and that was fun, actually. I was on the campus of Laval Université. And then, next week going, and then the following week, Susan Weiss is doing a symposium in Philadelphia. So we're going to do a TWiV there called The Origin of SARS-CoV-2. And we have her and me and Rick Bushman and this guy who's a retired FBI agent.
Oh, cool.
He worked for the FBI for 25 years, and he said he can tell us what he thinks they have because he doesn't know. He's not been working on it. But that's
right. Yeah, this is recall some time ago that there was this the Department of Energy, and I guess the FBI are the other ones who decided that they leaned or tilted towards the lab leak thing, right? And I have an acquaintance who has dealt extensively with the Department of Energy over his career. And I met with him not too long ago and, commented that it would be nice to see the data and what the Department of Energy had that made them lean that way. And he said, "You never will." Okay.
Yeah. And I want to just tell me, what does it mean, low confidence? What does that mean, right? So, and then after that is ASM in Houston, and after that is ASV, and then I think that's it for a while until August. Wow. And the fall is very busy. I got invited to Hong Kong and Australia.
Wow.
Okay.
They want me to go to Australia for 3 weeks and tour Sydney, Melbourne, and Brisbane.
That sounds like an opportunity.
If you're going there, you might as well go for a few weeks because you're going to be just over your jet lag after a week.
I once went for 5 days and came back. It was okay. I went to Singapore for 4 days,
That's rough.
Then I have 2 trips to Europe as well. So, a lot of travel. Might as well do it while I can, right?
Mm-hmm.
What the hell? Quebec, Angela, was very nice. If you go into the old part of the city, it's very nice. And people in Quebec are very friendly. And, they speak French. But if you don't, most of them speak English. And they're very happy to talk to you. So I had a good time. I was only there for 1 day. But
it's beautiful, especially now in the spring with all of the flowers and the trees and everything. It's gorgeous.
So, who was there, Rich? Jim Van Etten.
Oh, cool.
So we were he's
still upright. Huh?
He's upright, and we were looking for a restaurant on Friday night. And there are a lot of stairs in the old town, right? And he was just walking up the stairs, and we're like, "You want us to stop and wait?" He says, "No, it's okay. It's okay." And also Dave Dunnigan was there. He's the guy who says viruses get eaten by protists.
Yeah. What kind of, this sounds like some sort of DNA virus meeting or something.
Aquatic viruses.
Oh, okay. Yeah, Van Etten and Dunnigan have been, sort of lab buddies for decades.
How many viruses did they say there were per milliliter of ocean water? I remember there was a number that you guys had said. It was like millions. I believe it was in the millions.
Yeah, it's millions.
Which is crazy when you think about it.
It's funny. So the one guy I had on, Jed Furman, very interesting guy. He's all about systems readjusting, right? Because you take something out, he said the system will readjust. And I said, well, what if you could take all the viruses out of the ocean? He said it would readjust.
I find this, I think about this all the time, and I find it very comforting when I think about, what humans are doing to the planet. And, if we manage to extinguish ourselves or nearly extinguish ourselves, DNA will continue to replicate, which is good.
Yeah.
RNA too. But,
so anyway, after the podcast was over, Curtis Suttle came up to the podium to make some announcement. And he said, "Vincent, if you want somebody to come on TWiV and say, if you take all the viruses out of the ocean, life will cease, I'll do it."
Well, does this give hope for if we got rid of all of the mosquitoes, things would readjust?
Hey, listen, they may readjust and introduce something even more obnoxious than the mosquitoes. So,
it really depends which biologist you talk to, right? But this guy, I haven't ever heard someone so much into readjusting, right? I mean, he said, without viruses, the ocean would look like a log culture of L broth, right? It would be really dense with bacteria. And if you didn't listen to the episode, this is the last thing I'll say about this. He said years ago, something was eating the protists, right? There's a natural turnover of protists being preyed upon, but it was way more than made sense. And he said that's why we looked for viruses in the oceans, because we wanted to see if viruses were killing them. And in fact, that's what was doing it, because he said it was just too much of a turnover to just be other things. I didn't know that. That's pretty cool. Mm-hmm. All right. Now, Kathy, it's been a while since we talked about ASV, and it's not too far, right? What's up?
Right. It's really exciting. We have over 1,700 people registered so far. So we're looking at a record probably. We don't know how many will register between now and then in person also. But so definitely need to remind you, if you were at ASV last year, find your black neck pouch and bring it along with you, because we're trying to help save the planet a little bit and be sustainable with respect to the name tag lanyards. And you can help me win a bet with Andrea as to how many people will remember to bring theirs back. We will have them for people who don't or who weren't at Madison, but we're trying to.
How would you know how many to order, Kathy? How would you?
Well, we have basically the whole meeting covered again. Last year we ordered a giant order, and I thought it was going to be enough, and then people got nervous, so we ordered more because they think that people aren't going to bring theirs back.
No.
So
okay.
we have
I got mine. Mine's ready.
We have a nickel bet. Okay. And then we had told you about the showing of the film Virulent, and we are going to show it a second time. Actually, it'll come first in the program. We'll show it on Monday night. You would have to miss a half hour of the poster session. And then we'll also show it on Tuesday night. So that's pretty exciting. ASV is all about making choices. So we want to remind people, especially if you've never been in an ASV, there are concurrent workshops where there are 9 and sometimes 10 workshops at a time. And unless you have cloned 9 copies of yourself, you can't be everywhere at once. So you have to make choices. Same will be true in the evenings for the film versus the dance, or the film versus part of the poster session, or the social hour, etc. Anyway, and then the best piece of news is that we have the band to tell you about. The Magic Tones are an Athens-based party band. They perform at private and corporate events around Georgia and the greater southeastern United States. They feature members of a well-known touring cover band, Abbey Road Live, and another cover band, Cosmic Charlie. They've played at the New Year's Eve Peach Drop in Atlanta and a wide range of events, including at the Classic Center in Athens, where they will be playing for our event at the Pavilion, which is an outdoor roofed venue. So that's exciting.
Why do you think there's so many people? Because of the venue this year?
The venue, it's about, I don't know if it's going to be more than last year in the final numbers, but we have more abstracts. I think people are just hungry to come to meetings.
Travel
and
restrictions are all gone. It's time to get out again. Yeah.
This is the social pandemic-related equivalent of the baby boom.
Yes.
Right. So, yeah.
Yeah, and we will be doing a TWiV there. So
Yes.
Tuesday at noon, right?
Mm-hmm.
And I'm not going to tell you who's going to be our guests. You're just going to have to go.
Right.
All right. We have a research assistant position available at the FDA in Amy Rosenfeld's lab. I have to say I was there Wednesday to show a high school class a lab. And so I went to see it's very pretty. It's a huge campus that they built, because they used to be on the NIH campus, the FDA, and then they outgrew it. So they put them out somewhere in Silver Spring, and it's a huge campus. It looks like a university. So many buildings, all modern buildings. Anyway, so you could work there, and most of the buildings actually don't have labs in them. It's only one building, 75, that has some labs in it. The rest are people sitting at desks reviewing applications from companies to make products.
All those vast quantities of data that get submitted for a new drug or device or,
I mean, I walk past offices, everybody's got 2 huge monitors and then stacks of paper. Oh my God.
Oh my God.
You know, and the thing that's interesting, you only have to come in 1 day a week. You can work at home. So those buildings are all empty most of the time. Anyway, you could work.
But for the lab, you have to come in more often than that.
For the lab, you should come in every day.
Yes.
And you could work on enteroviruses, and you could work on making animal models. You could work on immune cross-reactivity. It's all BSL-2 work. It's a very nice lab. We brought the high school class there. So let me tell you, there's a high school in Washington, D.C. called Chance Academy. It's founded by Anna Bernanke, the wife of Ben. And I've gotten to know her. We share a fundraiser, okay? And she said, I want to bring my class to a lab. Do you think we could get into the NIH? I said, the hell with the NIH. Go to the FDA. You know, Amy will host you. So we brought him there. And so I gave him a little lecture on viruses, and Amy gave him a tour of the lab. And they all had questions made up ahead of time. It was very cool. So anyway, the labs are very nice, and you could work in one. So we will have a link to that position in the show notes.
And you could live in Maryland, hon.
Maryland. Is that from a movie?
No, that's just the Maryland accent.
It's very nice. Big, wide open spaces, lots of grass. They have outdoor areas where you can sit on the tables and stuff. But only one building has labs. It's just, that's CBER, Center for Biologics Evaluation and Review. So everyone has, even if you have a lab, you have to do a little review. And
you have to do a lot of review, I think.
Well, I better not say anything. Because every time I say something, I get, you shouldn't have said that. So I'm not going to say anything. Yeah, you have to do a lot of review. And I met my old postdoc, Gerardo Kaplan. He works there.
Oh.
You remember Gerardo?
Yeah.
He was at there. He was at
I didn't overlap with him, but I met him a few times when he came back.
He actually came to the polio symposium on Wednesday. Chumakov has so many interesting stories. You know what he said in his talk? This is the last diversion. Well, it won't be the last.
No, it's not.
Oh, no, this won't be the last digression.
He at the end of his symposium, a day-long thing, he got up and gave a little speech. And he said, you know, I don't want to say anything bad about the Soviet Union or anything good, but they pretended to pay us and we pretended to work. I thought that was funny, right? We do have science for you today. We have, and it just turned out that both papers have to do with influenza virus. I actually didn't plan it that way, but
well, it's a timely topic.
It is timely. And one is a review article. So I wanted to talk about high pathogenic avian influenza virus because many people are interested in this. So I found in Eurosurveillance, Avian Influenza, New Aspects of an Old Threat by Cornelia Aldhock and Francesca Baldinelli from the European CDC in Stockholm and the European Food Safety Authority in Parma. And so let me give you a little introduction to avian influenza. I actually talked about this at the Cosmos Club the other night in terms of the Fushe-Kawaoka ferret transmission experiments. Remember those?
Oh
yeah.
And when was that? That was like decades ago.
That was 2011. It started in 2011.
Wow.
Okay.
Oh my gosh. I mean, I dug up this talk I used to give for my class. Anyway, so avian influenza, you know, the reservoir of avian influenza A virus is birds, ducks, geese, swans, gulls, terns, shorebirds. Alan, did you go to yeah, you did. You went to the veterinary school in Tufts where we did a TWiV.
Yes.
Remember that? They work on these things.
Mm-hmm.
In birds, the virus reproduces in the gastrointestinal epithelium. It's shed in feces, little bit of replication in the respiratory tract, and it's often asymptomatic in aquatic birds, except for H5N1, which will kill them.
And if I were a virus and I wanted to get around, I can't think of any better way
That's right.
than
Bird
replicating in the
poo.
gastrointestinal tract of a migratory bird.
Yeah.
Oh, yeah.
So
You're going to get scattered on every statue between here and the destination.
I have a picture of a car that I show in my lecture, parked on a street, covered with bird poop, totally covered with bird poop. It's parked under a tree. Avian influenza is classified based on pathogenicity in chickens. There are low pathogenicity viruses, mild respiratory disease. They decrease egg production, so you don't want them. And then there's the high pathogenicity like H5 and H7. And we only have seen outbreaks of high path flu since 1959. The first one was in Scotland. And since then, more and more chickens, turkeys, etc.
Yeah, so high path flu is defined as killing
lethal,
farmed birds, right?
Correct.
Right.
Yeah.
And
Domestic
also,
poultry, yeah.
it has basic amino acids at the HA cleavage site, right? So that makes the HA cleavable by proteases that are in many tissues. So they tend to be able to replicate in many tissues outside of the respiratory tract.
Right. Okay.
Now, this is very interesting. The first outbreak of H5N1, high path H5N1, was in the live bird markets in Hong Kong in 1997. You know, they have bird markets. We actually have a bird market, many chicken markets here in New York. Did you know? You can go to one of them and pick out your chicken. They're still
Are you allowed
there.
to have backyard poultry or fowl in New York? I thought that was illegal.
I don't know about Manhattan.
I don't know about in the city.
But in the suburbs, you
certainly
can.
in the suburbs, I'm pretty sure.
Yeah, you can have it in
allowed.
the suburbs. So the virus that caused that was a reassortant. It was a new virus that arose by reassortment, and then it went into people. There were 18 human infections and 6 deaths, the first time ever for avian influenza. So they killed all the poultry, and that eliminated the spread. But then in 2002, it came back, and waterfowl then began to die. Okay, so that had never happened before. There was an outbreak in 2005 in wild birds in Qinghai Lake, China. There's a very famous outbreak. And then since then, it's spread all over the place. And now, of course, we have this really big outbreak, which is documented in this paper.
Since we're going to be doing both this and a neuraminidase paper, let's quickly bring the
talk about flu
audience
virus
up
life
to
cycle.
speed on hemagglutinin and neuraminidase and subtypes and that kind of stuff. So these hemagglutinin and neuraminidase are 2 proteins that stick out of the surface of the virion. It's a segmented RNA, negative sense RNA, membrane virus. It's got 2 proteins that stick out of the membrane. One's called hemagglutinin, the other's called neuraminidase. The hemagglutinin is essentially functionally equivalent to the spike protein in SARS-CoV-2. For those of you who joined us during SARS-CoV-2, that is, it is the virus attachment protein. So it attaches the virus to cells and helps it get taken up. Neuraminidase, we can do this more thoroughly later on, but has a different activity. It actually destroys the receptor that is used by the hemagglutinin. So it looks to be sort of nonsense, but that actually helps the virus spread, and it helps it get it through a mucus layer as well. And because they're on the surface, much the same as the SARS-CoV-2 antigens, they are immunodominant proteins. That is, when you make an immune response to the virus, it tends to be to these things, okay? And they vary significantly. Matter of fact, the hemagglutinin in particular varies all over the place, and it can do so because it's got a bunch of spinach, that is amino acid epitopes surrounding the actual active site that can vary a lot and distract the antibodies, okay, without compromising the function of the protein. And so there are many different types of the hemagglutinin out there that fall into, essentially non-cross-reactive subtypes. How many are there now? 18, or are there more?
18. Yeah.
18. And how many neuraminidase subtypes? 9 or 10, something like that. At any rate, the viruses wind up being described by what subtype of either hemagglutinin or neuraminidase they have on them. Okay, so an H1N3 influenza would have the
11.
Type
11, you're right.
Okay, 11 would have the type 1 hemagglutinin and the type 3 neuraminidase. All of these exist in birds, right?
That's right.
But
except
only,
the last
some are in bats.
The last 2 are in bats. Yeah.
Okay.
Yeah.
But only 3 of the hemagglutinin subtypes have shown up regularly as transmissible in humans, H1, right. H2, H3, correct?
That's correct.
And I don't know how many of the neuraminidases, but certainly not all of them. And so...
N1 and N2.
Okay.
N1, N2, H1, N1, H3, N2, N1 and N2 only, yeah.
Now, if somebody else can comment on whether or not the fact that the others don't show up in humans is because of the hemagglutinins or some other characteristic of the viruses that carry those hemagglutinins. Okay, there's all sorts of components in host range, but the fact is that only H1 through H3 regularly show up in humans and are transmitted, and the neuraminidase we already went over. So when we talk about this avian influenza as being what, H5?
High path, H5 and H7, yeah.
Right. Those are influenzas that don't ordinarily show up in or transmit in humans, okay? They can infect humans, as we'll probably discuss, on occasion, and with varying consequences, but are not regularly transmitted. But they're really, as we'll discuss, nasty in birds, this one in particular that's come up. And so, the concern is, what if?
Yeah.
Of course, we ought to be concerned about the birds too, right, Angela? Okay.
Of course.
But the other concern is, what if they got into humans and turned out to be transmissible, which takes us back to the Kawaoka experiments, okay? Yeah. I mean, we just went through a pandemic, folks. Yeah. All right.
Mm-hmm. And the sort of flu's evil genius twist on all this is, so you can combine different H's and N's, but flu, because it has a segmented genome, as Rich mentioned, if you get any 2 flu viruses in a host, they can potentially swap pieces of their genomes and you can get these new combinations. And so we're constantly rolling the dice. Whenever poultry get infected with an H5N1, they could also be infected with an H3 something, and they could reassort, and you could get something that comes out. Or a raccoon gets infected with the H5, and you reassort there. So the concern is there's all this stuff going on, all this exchange going on, and it keeps happening, and so we need to keep an eye on this and try and get ahead of it.
And that reassortment is not a low probability event. If you have a cell co-infected, they are going to reassort, and give you all possible combinations out the other end.
Okay, I want to jump in and add a couple of things. So the hemagglutinin, which is shortened to H when we talk about H5N1, is also shortened to HA for hemagglutinin. And the neuraminidase, which gets shortened to N, also gets shortened to neuraminidase NA. And there was an allusion to this that because one, the hemagglutinin binds the receptor and the other destroys the receptor, it's really quite a paradox. And if you think about it, Vincent and I like to teach this. I know that it's really important that there be a balance. And if a virus has a mutated hemagglutinin such that its ability to bind to the receptor is altered, then the neuraminidase in that virus ordinarily will have to change to accommodate that change of the hemagglutinin binding. So it's a really important balance between those two.
Interesting. I never thought of it that way. That's cool.
And so what's been going on with avian influenza most recently, which I think is really interesting, is exactly this ecology, because there's been avian influenza in poultry flocks, as Vincent said, for years and years and years. And as this review, which is open access, talks about, this is kind of an annual thing, right? In poultry flocks in Europe. And my understanding of the poultry industry is it's manageable because chickens that are being raised for meat are not around for more than a year. They hatch, they grow, they get cooked. So that's not a problem. They clear everything out. But what's happened just since 2020 is that this whole dynamic has changed, and we've got the migratory birds spreading it worldwide, and it has vastly expanded.
Yeah, so since 2020, there have been huge epidemics in Europe, 14,000 outbreaks, and people had to cull 96 million farmed birds. And they say, there have been changes in epidemiology that have resulted in this. As Alan said, the high-path influenza used to be seasonal. And what happened was migratory waterfowl would return to their sites in Europe where they overwinter in the autumn, and that would spill over into farm birds, and you'd get outbreaks. And then for the first time in 2021, there were high-path influenza viruses in wild birds over the summer at some breeding areas in Northern Europe. And then in the fall of 2022, huge outbreaks in the winter and spring that continued in the summer months. So something changed, and we don't know what it is.
Yeah, and I can't help but think the timing is pretty suspicious, that this was right when all the travel got shut down and a whole bunch of other things got changed, and the supply chain for poultry may have changed in some subtle ways.
Maybe.
And maybe that has something to do with this change in the virus dynamic that got it into waterfowl that were around all year.
Yeah, the seasonality changed, and that made a big difference. Yeah. And so it was also spread. So infected birds have now been detected in countries that never had it before, like Iceland, Norway, as far north as Svalbard and Greenland, all the way down to the tip of Chile. Never been sawn before.
So it crossed into the Americas and made it all the way through the Americas.
Yeah. Now they speculate that this persistence during the summer months may have happened because these viruses reassorted with low pathogenic viruses, right? So now they could infect new hosts. And they say there were some rarely affected bird species like barnacle geese or seabirds. And so that brings the virus into new populations. And they say there were massive die-offs in colony breeding seabirds in Europe and South America. Over 40% of the pelican population was killed in Chile and Peru because of this virus. And they say they're already endangered, these pelicans, and penguins are also at risk.
Man, penguins. Jeez.
Yeah.
All right, so that's birds, but as you've probably heard, it's also been infecting both free-living and captive mammalian species. And they say there have been a number of individual animals, like carnivores preying on infected, sick, or dead birds, but also marine mammals have been found in Europe and in the Americas along the coasts. They have had mass mortality events and seals, and then on land farmed minks, and then sea lions.
Seals have a lot of contact with seabird guano.
Yeah.
Like a lot. Mm. They're on islands that are made of guano and that are migratory stops, and so that's not that surprising.
So this
but
is
some
unusual.
Of these others are further inland.
Infecting mammals. So less than 1,000 human infections have been documented over the years, but this is a lot of new mammals being infected. So that's a bit of a concern. I asked Marion Koopmans in Switzerland whether it's transmitting among these minks and sea lions. She said it's very hard to figure that out, even though the paper suggests it. She said she's not convinced that it's transmitting yet. But there is also other, for example, they say infected, dead, or sick birds and foxes have in urban and peri-urban areas have led to increased environmental contamination, and your pet could get infected, right, as a consequence. Let's see. Yeah, so there have been some human infections associated with this, but not as many as you might think, given the extent of circulation in birds.
So we're talking about human infections associated with this particular virus.
Yeah, this particular one. Yeah.
But there have been, if I'm not mistaken, serosurveys in the past to try and determine the extent to which these other than H1 through H3 subtypes have infected humans. And human encounters with these other subtypes are much more frequent than you might have anticipated based on the serology. Okay. I mean, don't necessarily get sick or not sick enough to wind up reporting it as an odd flu infection. But my bottom line would be that this virus is testing itself against all sorts of species all the time.
Yeah, there were serosurveys done in countries where people farm birds, right? They have a lot of contact, and they had a few percent seropositivity. And so the human, the known human infections where someone gets sick, as I said, less than 1,000, but the fatality is about 50% in those. Compared to less than 1% for a seasonal influenza.
So let me be the devil for a minute here. Wouldn't you like to know what it is about these viruses that allows them to infect or transmit other species? Wouldn't you like to know what it is about these viruses that might allow them to infect and transmit in humans? Okay. And how would you construct such an experiment?
Gee, so I would take a ferret, which is a good animal model for influenza.
Maybe passage these viruses in potentially susceptible mammals. Yes.
And analyze the results.
Right.
No. You can't do that. That's gain of function.
So one of the things, this was a topic of last year's TWiV at ASV. What was his name? Who said?
Andy Bailey.
Andy Bailey was talking about the kinds of changes you need for influenza viruses to go from birds to people, and they're saying in this little review article, one of them is a change in the hemagglutinin, so that it now recognizes a human sialic acid and not a bird sialic acid, if you will. And they're seeing some of these changes in these mammal infections, but they're saying that we only look at dead animals, basically. We need to have more surveillance.
Yeah.
Of, I was talking to Countess Subarau in Switzerland. I said, we have all these people who they don't want us to do surveillance in bats because they think it's too dangerous for coronaviruses, right? But don't you do that all the time for influenza? She said, of course we do. We can't have a surveillance system without it where we're going to know what vaccine to use. And so here too, for the H5 viruses, you have to do surveillance. Can't put your head in the sand.
But do you think part of that is also because there are some quite severe hemorrhagic fevers that have been described in bats, which are potentially more dangerous than other orthomyxoviruses, like other influenza viruses? But they're still dangerous. I'm not saying they're not. Just there are certain
maybe,
like
but
BSL-4 pathogens that could be found in certain bat species.
I think you can't be scared, right? You have to.
Oh no, of course not. I would.
But maybe that's what the ferret transmission experiments. That's one of the things that people were scared that these viruses would get out and transmit among humans, right? I showed some of the headlines that were published at the Cosmos Club, and one of them was scientists describe 5 mutations that allow H5N1 to spread in humans. Right? This is the kind of
we wish we
lack
could
of understanding. And of course, and I put up a quote by Tony Fauci, and this is something that we harped on all the time. He said, a common misconception was that the aerosol transmitting H5N1 were as virulent as the original. That's not the case. And they lost their pathogenicity for ferrets, which nobody paid any attention to, if you remember. Anyway, so in this paper, they say that, the problem with going into mammals is that they're going to reassort. The H5N1s are going to reassort with mammalian influenza viruses. Then you may make something that can transmit. So you really have to do a lot of surveillance, which we're not doing. And they say we need to do more of looking and seeing what's out there, which I agree with. But the good news is that we have some H5 vaccines that have been stockpiled that could be useful. And we have the antivirals, influenza antivirals work on H5N1 subtypes. So it's not like the beginning of COVID, we had nothing, right?
So are these highly pathogenic influenzas that show up in domestic poultry, are they all H5 or are there other hemagglutinin-bearing flus that show up that display this kind of phenotype?
Sometimes H7s, but mainly H5s. Yeah.
Okay.
Yeah. So when I was in New Hampshire, I don't know if I told this story. I met with a veterinary microbiologist, and he said, they help the surrounding farms. And he said there was a farm up north. It was a mom and pop farm. They had 100 chickens, right? And they called and they said, our chickens look sick. And he said, okay, we'll send someone to swab them. And by the time they got there, they were all dead. And he said they had a little lake on their property, and the ducks poop in the lake, and the poop has virus in it, and it spreads to their chickens. And that's it. And they all died. He said it's amazing. It just wipes out domestic poultry.
Well, that's why domestic poultry normally is, at least at an intensive scale, the chickens never go outside. And if they go outside, they're very protected and they're kept away from wild birds normally.
Yeah.
Because of this reason specifically, right? Like if they're able to roam freely and come in contact with these sources of water. And
so that's the thing. It was a mom and pop operation, so they didn't have.
Yeah,
so
that's unfortunate.
Angela, do you think people shouldn't have bird feeders? Do you think they'll attract wild birds that might have high path?
Bird feeders that would be attracting, I mean, if they're attracting sparrows and things like that, or smaller birds, not necessarily ducks or aquatic wild birds.
Pelicans.
Pelicans, yeah, those sorts of birds won't really be sitting on a little bird feeder.
Just flying off with the bird feeder in the mouth. I can picture it.
They would be stymied by the anti-squirrel bird feeders.
Actually, I was out on the river last week, I think it was, here in Austin, and a pelican flew by. And I thought to myself, dude, you're lost. Okay.
Really? It is?
Yeah.
Wow.
Huh. Well, so yeah, if it's just sparrows and woodpeckers, I'm not aware of them getting infected, right?
No, we've never heard of any of them getting infected with or having highly pathogenic influenza
How about
viruses.
wild turkeys?
Turkeys, I think, actually are.
Yeah.
I think I actually read wild turkeys. Yes, they can be.
We have a lot of them in Massachusetts.
What's a barnacle goose? Is that the kind of goose that walks around in the backyard? The barnacle goose?
It's a goose that sits still for too long in the water.
I don't actually know.
Barnacle geese. Because the FDA campus was full of geese.
I've never seen this goose before. It's kind of like small and black with a white head. It looks nothing like the Canadian goose, which is what we have around here. It looks quite small, actually.
And the problem with a barnacled goose is you can't put anti-fouling paint on it. Sorry, boat joke.
So what's the bottom line on this article? That there is an H5 avian influenza going around that is spreading more than it used to, both in the natural host, and it's also spreading for longer. That is, it's not showing the same sort of seasonality as it used to. Spreading more in the natural host, spreading more in hosts that are more incidental, that is, other species of bird and mammal, and causing, probably as a consequence, is it fair to say, as a consequence of that, more problem in domestic fowl as well? So the birds here are experiencing their own little bit of a flu pandemic.
Yeah, when you said what's the bottom line, I thought be aware, be very aware.
Yeah, definitely. If you see any dead birds or other mammals, keep your pets away from them. Don't get near them. Obviously, if you have them in the forest on leash or near large bodies of water where there could be many birds, or bird feces, goose, which a lot of you guys also have the Canada geese hang around the northern United States. Yeah. And I'd like to add, actually, now just talking about a veterinary tool that we use, which is more of a One Health approach to identifying outbreaks, which is the World Health Organization equivalent in the veterinary field, which is the World Organization for Animal Health, which is also it was previously known as the OIE. And it has over 182 delegates worldwide. So there's veterinary representatives worldwide, where basically it assures international safe trade of animals and animal products between different countries, and also defines disease status in each country, looking at early detection, notification, and control of different zoonotic outbreaks. And there's a tool actually called WAHIS, which is the World Animal Health Information System, where it's actually this really cool map, this interactive map, where basically it's updated daily, where you can consult different outbreaks that are ongoing, or some of them are called disease events. There's specific definitions for outbreak versus disease event, but I added a little picture of today's where we can see that there was one African swine fever virus was identified in Russia on June 2nd. So there's quite a few actually for today for influenza A viruses, like in Canada here, it's H5N5, I believe. And then also in Russia, H5N1, Guinea, H5N1, Russia, also another account of H5N1. So it's just a really good tool, which is actually available to anybody, not only public health officials or veterinary officials, but anybody that wants to go onto this website, WAHIS, part of the World Organization of Animal Health. You can look into this. It also has all of the reference laboratories and collaborating centers, and all of the guidelines, diagnostic tools, and everything that are used, that are harmonized through all of the countries for detection of said pathogen. So I wanted to put that out there as a very, very important tool in preventing outbreaks, like things with influenza H5N1.
Very cool.
Case.
And in preventing outbreaks, the organization is WHOA. Yeah.
There's a case of BSE in the U.S. I'm trying to find it here. March. Oh yeah. March. Yeah. May 15th. Bovine spongiform recurrence of an eradicated disease. What is that about? Wow. Eradicated. Anyway.
Well, because it's considered eradicated in the United States, I believe. It's not considered active. So there's different statuses depending on the amount of the outbreaks in a certain country within X amount of years. They'll consider it either eradicated or like near eradication. So if a BSE, bovine spongiform encephalopathy, hasn't been seen in, let's say, 10 years in the United States, and there's an outbreak, it's kind of put on like a caution, like watch out list.
There have been more recent. Yeah, they occur sporadically in cattle in the U.S. and Canada too. Unless they're shown as,
I think there's different forms of the prions, right? Like depending on what, like BSE is a general term, but if they're not seen as transmissible or in more than one host, because they're seen sporadically in individuals, and also in small ruminants, which they call scrapie.
This was in Tennessee. Probably a cow just, sporadically came down with it because you have to. They have to be over 5 years old, and many cows are slaughtered before then, so you never see them. But if you keep a cow a long time, say for milk production or something like
dairy cow,
you would see it. But
when I was in Spain, actually, I had to. Everyone in the veterinary degree has to pass through the slaughterhouse. It's part of the degree, and every single cow that it's just part of it, because all of the animal welfare and also the way that they're euthanized is all very controlled. And we actually take from every single cow that is slaughtered, part of their central nervous system is then sent for sampling to make sure that there's no BSE or any other form of transmissible.
Where is this, in Spain?
This is in Spain, but in Canada, it's actually the same. There's also testing for bovine spongiform encephalopathies.
So every cow that's slaughtered is tested?
So in Canada, I'm not sure it's every cow. In Spain, I know it is every cow. In Spain, for sure.
In Japan, it is every cow also. They only have 10, so it doesn't matter. They charge so much for them.
They
cows
charge
are $1 million.
$10,000 for a steak. Yeah.
Wagyu beef. But in the U.S., they don't check every cow. That's part of the problem. You know?
So, Angela,
I think also the volume of cattle in the U.S. is much larger than it is in Spain.
That's true.
Does this organization have a headquarters, and who's in charge, and how do they fund themselves?
The headquarters, I think it's in Europe. Let me remember where in Europe.
I see a bunch of different branches: Africa, Americas, Asia, and the Pacific, Europe, Middle East. So it's global.
But there is one, I believe, in Europe. I just don't remember right now where. And I think the funding is via the 182 delegates. Each country contributes proportionally. But I don't remember actually where. I can Google it quickly.
Wiki, man.
I'm on their corporate website now.
Oh, it is in Switzerland. It is in Switzerland. No, that's the WHO. Sorry, that's World Health Organization. No. Animal World Health.
WO gives a contact address in Paris.
Okay. Oh, yeah, it's true. Yes, headquarters are located in Paris. Yeah. Okay.
Yeah, I'd have to dig deeper to tell you about funding. It's probably something similar to WHO.
Cool. That's neat. Didn't know about that.
But the interactive map is really, and it's updated daily, so it's really interesting. And these is just anybody, any veterinarian that identifies an outbreak, that sends a sample for serological testing and identifies any pathogen that could be potentially zoonotic. Or not only zoonotic, but that could cause an outbreak even in domestic species or in wildlife. They can identify the other 182 countries.
They also have an overview of all the animals that have been infected with SARS-CoV-2 that have been reported to them since the beginning of the pandemic.
Yeah.
It's
a great tool. Everything, it's a wonderful tool.
Cool.
Neat.
It's interesting because I went to the Johns Hopkins dashboard yesterday. They stopped reporting since I don't know,
a couple months ago.
Yeah, they just stopped because there's not enough testing anymore. It's interesting. I remember we used to go look at that all the time. No more. All right, so that is avian influenza virus.
That was a snippet, folks.
That was a snippet. Now we go into hour 2 of the show with the paper.
It's not easy to figure out. If you're interested, obviously this epizootic, right? Is that the name for a big outbreak in animals, Angela? An epizootic? It's not easy to figure out why it's going on because people are quick to say it's mutations in the virus, but it's also environmental.
That doesn't answer the question.
It's environmental stuff.
Because the virus has been mutating for as long as it's been around. And the question is, why didn't we see this blossoming and year-round spread in the preceding few centuries?
No, I think potentially part of it, and this is just my hypothesis, or just a thought, that in the more we intensively farmed animals and we crowd them all together in these very, very small enclosures sometimes with poor ventilation, and these animals can be severely immunocompromised because they have poor diets. They're eating only corn, they're vitamin deficient, but because, like I believe Alan said before, their lifespan is sometimes only 60 to 100 days, they're not necessarily the healthiest, which is unfortunate to hear, but it's just the truth. And in that case, if they were to be confronted with, let's say, X virus, then they would be more susceptible potentially to getting sick. I mean, this is just a thought of stress induced. That they're under higher stress, they're in dirtier environments, especially like hens, laying hens, as opposed to broilers. Broilers are what you call like chickens for poultry, for meat consumption.
Yeah, for cooking.
Exactly. Their only maximum lifespan is 120 days, but some hens can actually live up to 3 years. And in that case, if they're in like unideal conditions for 3 years in very small enclosures, maybe that's it. Like if we think about 100 years ago, intensive farming didn't look nothing like it does today. So I'm not sure if that's one of the factors.
But I do think the timing is mighty suspicious. Right when the world shut down from COVID-19, immediately afterward, we saw this major ecological shift in avian flu while all our supply chains were, going haywire, including food.
For sure.
It seems like there was something in there that changed that caused this to
beware of associations.
Enter a new chapter. Yes,
for
beware
sure.
of a
Yes, for sure,
it
for
is a
sure.
correlation.
All right.
Yeah, we're just throwing out, we're speculating.
Yes, just hypothesizing.
How would you begin to address
test
it?
that.
It's very hard, right?
Yeah.
Very hard.
Well, now that it's everywhere, this gets very hard to deal with.
And also, it's so asymptomatic in fowl, right, in the wild. So you just have to do random sampling of aquatic fowl
exactly
because most
right.
of them are asymptomatic. You're not going to see a duck sneezing or a duck with severe respiratory. And if it would be extremely rare unless it has, H5N1 perhaps. But for sampling, it's difficult as well.
And they're not going to report themselves to the World
That's
Animal
what
Health.
they say in this review.
go to Paris.
We need to do more sampling because we're just looking at sick and dead animals. That's not good enough.
Exactly.
Yeah.
Exactly.
Okay, so in hour 2 of TWiV, I like that, Alan. In hour 2, our article is a pan-influenza.
Coming up next in the second hour, we'll be calling
a pan-influenza antibody inhibiting neuraminidase via receptor mimicry. 3 first authors: Corey
Four.
Corey Momont, Javi Dang, Fabrizia Zata, and Kevin Hauser. The last authors, Matteo Samuele Pizzuto. I have to say this other name, Antonio Lanzavecchia. And these are all over. They're from Vir Biotechnology, Humabs Biomed, University of Cambridge, University of Utah, and Washington University School of Medicine. And listen to this number, 300,000 to 600,000 deaths per year from influenza infections worldwide. I didn't know that number. That's big.
Humans. Now we're talking about humans.
That's
Yes.
humans,
So
yes.
humans,
We're not-
Yes.
Exactly.
Which could be underreported as well, right?
In the U.S., it's what, between 20 and 50,000 or
Yes,
something like that,
something like
depending on
that.
the year.
That's the figure people usually cite, yeah.
And this paper is about both influenza A and influenza B viruses. The influenza A viruses are the ones that are all throughout these wild birds that we talked about and bats. And of course, they can infect pigs as well. But influenza B viruses are mostly human viruses, but they can, we vaccinate against both because they can both cause severe disease. So we talked about the hemagglutinin, one of the spikes on the virus particle. It binds a receptor, which is sialic acid. Then the other spike is a neuraminidase that cleaves sialic acid from membranes, and it allows the viruses to move away from infected cells because these viruses are released by budding at the plasma membrane. So you could imagine they would just immediately stick to sialic acid receptors if the neuraminidase wasn't there to cleave it off.
I want to just sialic acid for a minute. Okay.
Yeah.
It's can you do that for me? It's like a carbohydrate, a sugar, right?
Yeah, it's a 6-carbon sugar. Yeah.
That is post-translationally added to numerous different proteins.
Yeah.
In a specific fashion. So cells, lots of cells have sialic acid all over them. Is that right?
Every cell has sialic acid.
Okay.
It's everywhere.
And so
Billions.
it is a ubiquitous receptor.
It is.
And the hemagglutinin is specific just for binding to that thing, regardless of what protein it's on, right?
Regardless of what protein, it binds sialic acid. And there are 2 kinds of sialic acid that it could bind to. So a sialic acid is a 6-carbon sugar with an acetyl group on it. And the acetyl group can be on different places. And for human influenza viruses, it's one of the sialic acids. It's called alpha-2,6. And then for birds, it's alpha-2,3. And then, so for a bird influenza virus to infect mammals, it has to be able to bind alpha-2,6 sialic acids. It's the first thing that has to happen. So this is all about, trying to prevent infections.
Right. And so just a brief, we've talked about hemagglutinin and neuraminidase, and these are the 2 main antigens on the surface, and they're, critical for the viral life cycle, and they vary a lot. And that's why you need a new flu shot every year, because as new strains come around, you need to vaccinate for the currently circulating antigens. So for a long time, people have been wondering if we could get a universal flu vaccine that would, instead of responding to these highly variable parts of hemagglutinin and neuraminidase, maybe respond to and bind the fixed regions, because there are parts of these proteins. The hemagglutinin has to have its enzymatic activity as a hemagglutinin. The neuraminidase has to have its enzymatic activity. There are very conserved regions of both proteins, but we don't usually generate antibodies against those conserved regions. If we could, then you'd get one flu shot and be done with the flu, maybe. So people have for a very long time been looking for antibodies that do hit conserved regions of these two proteins.
Hemagglutinin doesn't have an enzymatic activity.
Doesn't have enzymatic.
The neuraminidase does.
But it does have conserved regions.
So the idea is that the antibodies against hemagglutinin are really important for protection, right? But people have ignored the neuraminidase, but there is some evidence that neuraminidase antibodies are important as well. And but the problem is the vaccines that we use don't induce good antibodies to neuraminidase, right? So people have been trying to get around that. And I think there was recently a paper where they tried to use just the neuraminidase as an immunogen, or at least make more of it in the vaccines.
There's references to, at the end of this paper, there's a reference to neuraminidase vaccines. And I looked up a couple of those papers, and they're all about producing neuraminidase that are more stable. And then using those potentially as immunogens. And there's a fair amount of research that suggests that might be a productive pathway.
But this paper is about a monoclonal antibody that could be used therapeutically. And as Alan said, people have made monoclonals against the conserved parts of the hemagglutinin, thinking they could be good because the conserved parts don't change much, right? So maybe you don't have to get a vaccine every year. But they say here in therapeutic tests, most of these have no or limited efficacy. One of them is in a phase 2 prophylactic study. That's an anti-HA conserved region monoclonal antibody. So the focus of this paper is can we make monoclonal to neuraminidase and show that these could be useful, which would then, say, stimulate more research into putting neuraminidase in vaccines, maybe.
Right. Well, it would be twofold. I mean, the monoclonals are sort of, on the one hand, you could get a useful therapy right out of that. If you've got a monoclonal antibody that will neutralize a broad spectrum of flu viruses, then you could use that against whoever comes in with the flu. But it would also give you the kind of the direction to go with what kind of antibodies you want to induce with your vaccine.
They actually say here that the monoclonal that we're going to talk about, it looks really good, as you'll see. It could be a single dose per season prophylaxis against influenza. That would be interesting, right?
Yeah.
It's not a vaccine. It's a passive vaccine, right? Where you get a bolus of monoclonal and maybe it protects you for a flu season.
Yeah, because you can add these stabilizing structures to an antibody, put it in somebody, and it'll stick around for months circulating, and potentially, I assume that what is the company, Vir Biotechnology and their subsidiary Humabs are working on that plan.
Although
but the approach here, they just started off with sera from people, right?
Yeah, they screened sera from people for cross-reacting antibodies to neuraminidase. So they produced neuraminidase on cells, and they screened the sera for the ability to bind. They found one donor with high reactivity against the different neuraminidase. So they tested
donor I.
Donor I, yes.
And I don't know if they named them retroactively as like for influenza, or if it was, they just happened to be early in the alphabet when that donor showed up.
But then basically they looked for memory B cells from that donor using a neuraminidase, what they call a baiting strategy, an N1 neuraminidase. And then they get single cells, single memory B cells. They take the supernatant, which has antibody, and they screen it for inhibition of the enzymatic activity of neuraminidase, which, as we said, is a sialidase. It cuts off sialic acid from the glycoprotein. I love this assay. You've all heard of ELISAs, right? Enzyme-linked immunosorbent assay. Have you heard of ELA?
No, this is a new one.
Enzyme-linked lectin assay. Isn't that cool? ELA?
Oh, okay. So I've seen it before. I didn't know it was Ella.
I actually want to say under my umbrella, Ella. Ella. Remember that?
Rihanna, of course.
Rihanna, under my umbrella, Ella. Little did she know she was talking about a lectin assay. Anyway, they get a bunch.
Maybe she did know. I don't know.
They get 14 monoclonals, and three of them called FNI9, FNI17, and FNI19, bound to lots of different neuraminidases, including seasonal and animal circulating, both A and B. So that's what they studied further in this paper. Do
we know where the 200 healthy individuals were from? Does it say? It doesn't say which country, like where were they sampled?
I didn't. I was actually kind of surprised that it was only 200 people who they looked at, and they hit gold. And I don't know like what the background on
sample
that.
Donors, Swiss blood donation centers.
Okay, Switzerland.
Okay.
So these are clearly unusual individuals because they're from Switzerland, right?
It's expensive blood.
The Swiss blood donation centers, yeah.
Well, if you want to look for neutralizing antibodies, you go to Switzerland.
Yeah, very good. I just got it.
It took like 4 seconds to kick in. I was like, wait.
All right, so then they took these antibodies and they say, do they neutralize infectivity? Do they block infection? And they checked viruses spanning 80 years of neuraminidase evolution. And the three antibodies, FNI. What does FNI stand for?
We don't know. They never tell us.
They didn't say.
And I really had a lot of debate whether it's a 1 or an L or an I.
Because
I had to copy and paste it into another document in order to get a better font to see that it's an I.
It's an I.
Okay, good.
Okay.
It wasn't.
FNI.
It is a capital I.
Huh.
Yeah.
Interesting.
Unless maybe it's like one of the first authors.
All right. Well,
the patient, I.
Anyway, these three
usually
FNIs.
Usually, well, no, that wouldn't be the case. I was going to say usually monoclonal antibodies get labeled with what their number was on a microtiter plate. Okay,
yes.
But that they wouldn't all be FN if that were the case. This is
G32 or something. But and no, this is.
So these three monoclonals neutralize all of the viruses that they tested, right?
Mm-hmm.
Yep.
Yeah.
Bunch of A viruses, bunch of B viruses.
Bunch of B viruses,
yep.
Yeah. And interestingly, so some recent H3N2 influenza viruses have a new sugar on the neuraminidase, position 245. And this has been previously shown to interfere with antibodies against the neuraminidase. So they ask whether this makes a difference because it's a currently circulating virus, so that's relevant, right? If you're going to be treating people with this antibody. And it did decrease binding of these monoclonals, but some of them still had nanomolar binding affinity despite this sugar being present. So they say that they might still work against viruses with this N245 sugar added to it.
By the way, when we say neutralizes these viruses,
right?
If I'm not mistaken, these are all pseudoviruses. And I looked into this, and they're actually, I think, retroviruses with the N protein expressed on the surface. And they are
all pseudos. They're not.
There may be some real viruses in here somewhere. I'm looking at this, but they use pseudotypes extensively throughout this.
They say pseudoparticles is what they refer to.
Pseudoparticles.
Yeah.
Pseudoparticles.
This stuff I just told you, they say we checked these antibodies against a panel of seasonal IAV and IBV strains.
Okay, good.
I think that's the real thing.
Yeah, okay.
Yeah.
But the next set of experiments is using pseudoparticles with neuraminidase of enzootic and zoonotic avian and mammalian influenza A viruses. So maybe they couldn't get viruses for all the ones they wanted to check, right? So they just put the NA gene in.
So all of figure 2 are pseudoparticles, right?
Yeah.
Even part.
And by the way, this paper is also open access if you want to go, if people want to read along at home.
So these antibodies do really well against all these different neuraminidase. Broad inhibitory activity against the panel of pseudoparticles representing all of these neuraminidase from viruses circulating in multiple hosts. Let's see. Then they did a combination experiment where they combine. So they're now looking at FNI number 9, and they combine it with an anti-hemagglutinin antibody directed against the stem. All right, and
so this is one of these HA antibodies against a highly conserved region that people have been trying to develop as a potential antiviral and as a route to a universal vaccine.
And they synergize. They work more than additively. If you add the neuraminidase antibody and the hemagglutinin antibody, they do much better than adding. So that's a therapeutic possibility to use both of them, right? And they conclude that these neuraminidase targeting monoclonals have unprecedented breadth to seasonal and zoonotic influenza A and B viruses, and they can synergize with these HA stem antibodies. So then they do some structural analysis. They want to know how these antibodies are working. So they bind the antibodies to neuraminidase protein, and they solve the structures by cryo-electron microscopy.
And I just have to comment. It was another one of those. God, this field has really progressed. And I'm reading along, and they're saying we solved the structure by cryo-EM at 2.9 angstrom resolution. Supplementary table 5.
I was going to say it's a supplementary figure. Yeah.
It's just, by the way, we solved the structure of this, which, used to be like somebody's thesis. And
yeah. Well, cryo-EM
it's just on the way to the answer they want to get. And so that's often the supplementary.
In the old day, you had to do crystallography.
Yeah.
It was just so random whether you could get a crystal, whether it would diffract.
Yeah.
And cryo-EM has totally revolutionized. It's like, we used to sequence a viral genome and get a paper.
Yes.
Now you get the cryo-EM and it's a supplementary figure. Paper. Anyway, what they find is that the antibody, the region that binds the antigen, just called complementary determining region, it inserts into the enzyme active site of the neuraminidase and makes interactions with the residues that are part of the neuraminidase active site.
It effectively mimics a sialic acid.
That's right, they're saying. Yeah, it mimics the way the sialic acid would fit into there. Exactly right. So they call that receptor molecular mimicry. I guess that's right?
Yeah.
Mm-hmm.
Sure.
And that's what Tamiflu does. Tamiflu mimics sialic acid. It's structurally very close to sialic acid, and it fits in. Tamiflu inhibits neuraminidase, and it fits in the pocket.
This kind of blows my mind. It's a loop on this antibody protein structure that sticks into the active site. It's crazy.
It's all you need is a little loop.
Mm-hmm.
It's amazing.
And the Tamiflu is what they use in this set of experiments. It's the oseltamivir, right?
Yep.
It's the same thing.
It is.
Which is just like known as a neuraminidase inhibitor, right? Broadly.
Yeah.
It's defined as that. Yeah.
Yeah, it is. It was designed based on, it was a computationally designed drug based on the structure of sialic acid, right? So how close could we get to sialic acid and inhibit it? And it worked. You have two, actually. There's Tamiflu and Relenza is the other one. So these antibodies look like the sialic acid sticking in there. How about that? Isn't that amazing?
Crazy.
It is. And it suggests that it would be hard for the virus to get around
That's
that.
right.
Because you change the active site, now sialic acid doesn't fit and it's not going to work. And I was, as they did the experiment I expected next, where they looked for resistance mutations. And of course they find them, but it looks like it's costly.
Yeah.
These viruses are not in great shape when they change this.
Yeah, and they find that the changes are, so there's a huge database of changes in sequences of viruses you isolate from people, and they say these are very rare in human and animal isolates, suggesting there's a fitness cost for them, so they don't spread very readily in populations. So that's nice, right? And these changes, by the way, they don't completely destroy the ability of the monoclonal to work. They just reduce it, so it still works somewhat. But that's pretty cool. And they looked at the effect of these this sugar that is stuck on the neuraminidase of currently circulating H3N2 viruses, and what they do the structure of neuraminidases with or without the sugar. And without the sugar, you can see that so the neuraminidase is a tetramer, and you can see you can see up to 4 monoclonal antibodies bound to the tetramer, right? So each subunit of neuraminidase is going to bind 1 monoclonal antibody, and so that what they find is that with the sugar, you can see 3 or 4 monoclonal antibodies binding to them, whereas 0 or 1 antibody occupies the tetramer with the sugar. So I said it wrong. Without the sugar, you see 3 or 4 monoclonals binding, and then with the sugar, there's 0 or 1. So the sugar interferes with the binding of the monoclonal antibodies.
That's some of the monoclonals. Is that right? It says FNI9 is less affected by that. Mm. And this part confused me. I got lazy, actually. That's the truth. So this is what is this? A modification in a variation in the neuraminidase sequence that allows a residue to be glycosylated in some fashion.
That's correct. That's correct.
Which is, not an untypical type of variation that would affect the immunogenicity, blah, blah, blah.
That's right. That's right.
So we have to look out for this. All right. And that's why they're looking into this.
I got it wrong. Let me fix it. Okay. So there's more than 1 monoclonal. One of the monoclonals, you see 3 or 4 monoclonals bound in the presence of the sugar. And then the other one, you see 0 or 1. So there's a difference in the monoclonals being affected by the sugar being present. Right.
So that's one of the things that focuses through all of this. We start out with 3 monoclonals and we wind up thinking that one of them is the best. Right. And this is one of the criteria.
Right. The other thing they do here is they look at the structure of the monoclonal bound to the neuraminidase with or without the sugar. Okay. So without the sugar, there's some loops where the sugar is going to go. They don't clash with the antibody. They don't interfere with the antibody binding. But if this one antibody, FNI9, if there's a sugar present, there's a conformational change to avoid a clash between the antibody and the sugar. Okay? And they say this is very interesting. The energy required for this conformational change could be the fitness cost. That leads to lower affinity. I'd never thought of it that way, right? That in order to to avoid a clash, you need energy, and that impacts the affinity of the antibody bindings. It's interesting, right?
Yeah.
Ernie, what else would you like them to do? Would you like them to do an animal experiment?
Please.
Sure would.
All right. So then, with flu, if you're going to use mice, which is what they do, you're limited because not all flu viruses will reproduce in mice. So they use mice that can be infected with a specific strain of influenza virus, an H1N1 called PR8. And they make a murine-ized form of the antibody. Now, you've probably heard of making humanized forms of monoclonals to give people. Well, they want to give mice these monoclonals, but they're human monoclonals. And if you don't murine-ize them, which means make them mouse, they will be, you'll make antibodies against them in mice and they'll be clear.
Right. The constant region of the antibody that's from a human is going to alert the whole mouse immune system, and that's going to be wiped out.
Yeah.
So they made murine-ized versions. They also of FNI9, and they also make a change that will prevent complement binding. So they're looking at not just neutralization. It's a very cool experiment, but FC mediated activities, which would be complement binding. And they did the same thing. They murine-ized this HA stem antibody as well. So they do prophylactic administration. You inject the monoclonal intravenously 24 hours, then you give them a lethal challenge with influenza virus. The mice that got FNI9 were completely protected from loss of body weight down to a dose of 0.9 milligrams per kilogram, and with 0.3 mg per kg, limited loss of body weight. And all the animals recovered after 14 days. So this is a lethal dose, would kill the mice.
Yeah.
And the HA antibody also gives them full protection, but only down to 3 mg per kilogram. Right? The other one did 0.3, so much more active. And then they used the monoclonal with this single amino acid change that prevents complement binding, basically. And it still works. The neuraminidase antibody still works, but the flu and the HA antibody doesn't work as well. It's got limited activity. So they probably have two different ways of acting. One has more of an effector function than the other. They mixed these two antibodies together, and they work better mixed together, as we saw before with neutralization. And then they look at a panel of influenza A viruses and influenza B viruses, and they compare it to oseltamivir treatment. So if they give the mice daily oseltamivir, it reduces viral titers, but up to a certain point. And these are viral titers in the lung. But if they give the neuraminidase antibody, it reduces viral titers big time. And if you give more antibody, it reduces it even more. Even viruses with these sugars on them. And in contrast, the Tamiflu doesn't have activity against influenza B viruses, yet this neuraminidase monoclonal does. So in some cases,
it's a nice experiment.
It's cool,
a single dose.
Yeah.
They got plaque assays in here, Vincent.
They do. Influenza viruses are good for plaque assays. Now, I just said something I don't understand. We challenged mice with a panel of influenza A viruses and influenza B viruses. I didn't know that they would all reproduce in mice. I just finished telling you have to use this mouse-adapted strain PR8. I guess they picked viruses that would reproduce in mice, right?
Yeah.
I guess there must be mouse-adapted bees.
They've got it. Yeah, H3N2 Singapore and a B Victoria and a B Brisbane of Victoria lineage.
I guess they reproduce in mice. Okay.
I just have a little comment about this part of the figure because I wanted to try and match up the figure with the data that they're reporting in the text.
Right.
And these tiny little dots with these shadings that are very non-distinct between a pink and a peach.
Yes.
Oh, it's
And
terrible.
I blew it up to the full size of my monitor, and I still almost wasn't able to figure it out.
The color choices on these and the dot size are very unfortunate.
Yeah, they could have used different shapes or something. Yeah.
Yeah.
Yeah, that's
I mean,
Or
not,
put them in order at least, because
Well,
they're not even necessarily in order
Yeah, that's true. I mean, they are actually, now that I
from
see
the
it,
top. Actually, yes, from the top, yes. But if you look at G and H, I guess they're not.
then
but
they're
E and
not
F,
anymore.
yeah.
Right.
Yeah.
Yeah, it was very frustrating.
So at least in order there.
But
They are.
I mean, I have a really large monitor, and I figured that I was blowing it up at least 20-fold to see these things.
yeah,
Wow.
you're absolutely right. Those are so faint. Not a good choice. But also they're
Yeah.
green and red, which is not good for colorblind people, right?
True.
Right.
Yeah.
No, different shapes is a very good idea, Kathy.
Yeah.
Yeah.
And they have like the space to make these dots larger. They could have in
Exactly.
Prism
Yes.
or whatever they use, like increase it double,
Yeah.
and you would still have space.
Well,
Like
And in the legend down below, they could have made them
Yeah.
even bigger because they're all clustered. So you would be
Mhm.
able to figure out a color by cluster if the legend was big. It's hard, I know, when you're making a figure for nature and it's going to be small, but
So what we have here is a monoclonal against the neuraminidase, which can block infection with a wide range of influenza A and B viruses. It works prophylactically and therapeutically did they do therapeutic or just prophylactic? No, just prophylactic.
Just prophylactically.
It works in mice. And so the idea would be you could, as they say, you could give a single dose per season because, as Alan said, you can modify these monoclonals to last a long time. Or you could figure out how to make a vaccine that would induce these kinds of antibodies.
Yeah, that's what
right?
that to me is the real trick. I wonder how you would do that. Make a vaccine that would specifically induce this kind of weirdo antibody. Because after all, they had to screen
immune system.
200 people to find one person who did this.
Mm-hmm.
Subdominant, right?
Yeah.
Immunosubdominant. Same problem with the broadly neutralizing HIV antibodies, right? They're rare. I guess maybe they involve a lot of somatic hypermutation.
Oh, well, that's one thing they said about this donor. They pulled out a bunch of monoclonals out of this donor, and they were all related to each other and deviated by hypermutation. So, very interesting.
Rich, so you're in a company, and then your boss says, "I want you to make an immunogen that induces these broadly neutralizing antibodies." What would you do?
I'd call Brienne.
First, we need an immunologist.
I'd throw out a lifeline. I'm thinking you'd have to construct. I don't know. It's an immunogen that contains just very specifically the part of the or something that looks like the part of the neuraminidase that you want the reaction to. You can't use the whole neuraminidase. You have to just use something like the active site.
And you've got to hold it in the right configuration.
Actually, I heard a talk in Switzerland by
a scientist.
A scientist, a researcher at the conference.
The two-proline guy in Austin. What's his name?
Oh.
You don't remember either.
Jason McClellan.
Jason McClellan.
Good, thank you.
So they wanted to take just make the S2 part of the spike as an immunogen because it's really conserved, right? The S1 is at the top. That's where most of the neutralizing epitopes are, and they all vary, right? But the S1 is conserved. So he just produces S1, but it doesn't fold properly. So he's got to introduce all kinds of amino acid changes to make it open, and he does structures on each one and all this incredible engineering to make it look right. And then you put it in mice and then you don't get any neutralizing antibodies. I mean, he talked for 40 minutes on this and someone asked him, so what's the future of this? He said, there's no future. It doesn't work. But it's hard. It's really hard.
It's hard.
Can you imagine a person who did that work? But it's got to be done.
Probably more than one person.
Yeah, it was more than one person.
Definitely.
Anyway, so it's hard to get these conserved antibodies.
That's a
But
great example. A great example, though, of negative results that need to be published.
Sure.
Otherwise, some
Yes.
other poor clown's going to do the same thing.
Some
right?
other poor schmuck is going to get stuck on the same project. Yes.
Many years ago, we had a negative result and sent it to Nature, I think. And the guy, I talked to they just sent it back. So I called the guy. That's back in the day when you could call people on the phone. I said, what's it? He said, we don't publish negative results. Ridiculous. He said, well, that's just our choice, tough. And it got published in another good journal, high profile, but I think that's just to have a policy of not publishing negative results is not good because people need to know so they don't repeat it, right?
Right.
Of course.
And you learn things from what...
You learn things from what didn't work.
Sure.
Yeah.
That's absolutely right. That's the basis of how we live our lives, right? You learn from stuff that doesn't.
I learned from things that don't work all the time.
Yeah. All right. Let's do some picks, right? Because we are at the half-hour mark, so the picks will take us a half hour. So that's we have to do just one paper sometimes so we can get through some of these emails. Some of which are quite interesting. But for now, we will move on to some picks of the week. Let's start with Angela. What do you have for us? Okay,
so my pick is it came out today actually in Nature. It was just this really cool, basically there's the month's sharpest science shots as they define it by Nature. So Nature's photo team just chose a whole bunch of different striking images, and they're really cool. Like if you see them, there's one of a violet sea snail that builds rafts by trapping air bubbles in its mucus. And you can see this snail holding on to this raft of, as they call it, a raft of bubbles. And it actually, when I saw it, I thought it was a jellyfish because it kind of looks like a jellyfish. All of these bubbles bound in this mucus. Yeah. It's really interesting. They're really beautiful. And there's another one called yeast snowflakes. There's a picture of some yeast under the microscope. There's one actually we talked a lot about avian influenza today, where they saw that these northern gannets actually lost the color blue, the coloration in their eye was lost post flu, but they're not actually sure if it has anything to do with the virus or not, but that they saw that those that survived avian flu lost the color of their eyes. So obviously, I don't know if that's just anecdotal, but I mean, it was interesting. And there's other things, about platypus. Oh, one really interesting one is this guy, what's his name? Joseph Dituri has broken a record for the longest time living underwater, which was over 100 days.
Yes.
It's called Project Neptune. It's so cool. And there's a picture of him just in the window of his little submarine.
His habitat. Yep.
It's really fascinating. There's just a bunch of really fun pictures.
These are really cool.
Yeah, they're very cool.
Yeah.
So the yeast multicellularity picture is by William Ratcliffe's lab, and he I talked to him when I went to Georgia Tech in 2019. It's very cool.
Oh, cool.
De novo evolution of macroscopic multicellularity, and it said you have to come on TWiV, and then of course kind of got distracted, but I have to get back to him because this is a very cool story, and the pictures are great.
And the last one was really cool, the cold and moldy, where it's a picture of a mushroom, Pleurotus ernigi, and it shows this phenomena of fungal hypothermia, where you can see literally how this thermal image, you can see the fungus itself, and then around it, the heat, and how it's not really understood how fungi do this fungal hypothermia, but you can observe it here, this cold, you can see that the fungus is extremely cold, or the mushroom, and then its surroundings very, very hot. It's really cool.
I like the first one, which is the frog that can
the pollinating frog.
Oh, yes. Sorry, that was the main. Yes, that was the main photo. The pollinating frog. That was really cool.
Yeah, a Brazilian tree frog.
Brazil, exactly.
Zexon's Brazilian tree frog.
It's very pretty. Mm-hmm. Really great photos. Yeah.
Yeah.
And some of them, when you scroll, they change, right?
Mm-hmm.
Yes.
Yeah.
But the snail floating with the bubbles is really cool.
So cool.
Life is amazing.
Yeah.
We should stop trying to mess it up.
We're not trying to
No,
mess
we
it
don't.
up.
We just do.
We just are.
We just do.
Yeah.
Well, I think it's driven by profit, right, Alan?
Greed.
Yeah.
Greed, hatred, and delusion. The three poisons.
Greed, hatred, and delusion.
It covers the whole
I've
thing.
never heard of that.
Oh,
I've
yeah.
never heard that before.
It's only 2,500 years old. It's the three poisons that covers everything. Think about it. Okay, it covers everything pretty much in a non-overlapping fashion. And also think about the opposites of those: generosity, love, and wisdom. That's what we need.
I sat next to a guy on a plane a couple weeks ago. He was so strange. He was quiet, and then after we took off, he turns to me and he goes, "Is there anyone that you like?"
What, like, what does he mean?
Well, so I said, "Of course." What's the problem? He goes, "It's all about loneliness." And I said, "What are you talking about?" He was a financial guy from Toronto. Okay. One of these.
Oh no, a Canadian.
And he said,
nice.
all the problems of the world are due to loneliness. I said, no, it's money and power and sex. That's what I think. And Rich has his own theory. But he said,
Not
no.
mine, I
said,
copied it.
Are you telling me that Putin is lonely? He said, absolutely. I said, are you telling me Hitler was lonely? He said, absolutely. And he said, if people would just hug each other, and that's when I lost him. I said, okay.
No.
He said, I'm going to hug you when I get off this plane. I'm like, no, you're not.
No.
And fortunately, he got distracted and ran off. But he thought all the world's problems were due to loneliness.
Poor guy. He's probably lonely.
Yeah.
He's probably
all his problems may be due to loneliness.
He said Putin is very lonely and he lives by himself and doesn't have any meaningful interactions, and that's what drives him. I think sex, power, and money is better. But
and what was his opening question again?
Is there anyone that you like?
Is there anyone you
is
like?
There anyone that you like? Which is so weird, right?
Yeah.
That can be interpreted many ways. Like,
yes.
Is there anything you like? What do you mean? Like,
he didn't know me at all, right? So he's just
right. Well, that's what I wondered. Did he overhear you talking on the phone?
No, I wasn't.
Does he know anything about TWiV?
Nothing. He didn't even ask. He didn't even ask what I did, which is, when you get in a conversation with, and he talked to me the whole flight, which was just, I was from New Orleans to Dulles, okay? It was maybe 2-hour flight or something. And
That's very long.
He said that, you can tell me to shut up if you want. I said, no, I'm interested in hearing what you have to say. And then he got into a big rant about books. He said, tell me a great book you've read. Anyway, but the thing about loneliness, I ran it by some other people, and some people thought that it actually made sense, that despots can be lonely.
In some cases, probably, yeah.
But despots are not the source of all the world's problems.
I think they're increasingly becoming that. I mean, not the deforestation stuff, no, but
despots are often a symptom of the world's problems.
You think?
Yeah. Yeah, how do they get to power?
It's a con job.
Anyway. Yeah.
Anyway, I'm sorry. Kathy, what do you have for us?
Nice little rabbit hole there. Okay.
Yes. I picked an article that's a review article from November of 2021, and Justin sent me this. It's a really well-written historical review. The title is, What Were the Historical Reasons for the Resistance to Recognizing Airborne Transmission During the COVID-19 Pandemic? I didn't recognize any of the authors except the second one is Lindsay Marr, who we came to know as being the professor at Virginia Tech who is in civil and environmental engineering and knows a whole lot about airborne things and was found a lot in the popular press, but also in, I see Zeynep Tufekci on here also. Okay. Anyway, the article itself, it's open access. I highly recommend it. It's just a really nice historical thing. And the thesis is, for most of human history, the dominant paradigm was that many diseases were carried by the air. And this miasmatic paradigm was challenged in the mid to late 19th century with the rise of germ theory. And this is all pictured really well in Figure 2. And this germ theory sort of came about because of understanding how cholera and puerperal fever and malaria are spread. And so this whole paradigm shift that things aren't carried in the air, but maybe by the water, Jon Snow and the pump, or by hands or mosquitoes, really changed everything. And so the idea of air being bad became a very bad idea. And in particular, this one scientist, Chapin, really pushed that airborne transmission is crazy. And so that went away. And then until around 1961, when tuberculosis was really discovered to transmit through the air, and then later measles and chickenpox. But then really finally in 2020 with COVID-19, this paradigm shift back to, okay, maybe viruses or diseases can be transmitted by air. So it's like, why couldn't we have both all along?
A little balance here, maybe?
Yeah. And it's just fascinating and really well done. So it's not a very long read. Like I said, it's open access. It's got this great figure 2 that shows how everything used to be miasma, then there was a paradigm shift, then everything was not airborne, and now we're shifting back to, maybe it can be all of these.
Cool.
Yeah.
Neat.
Well, I think like in norovirus, which we typically consider fecal oral, there are cases where it's airborne. If you throw up, you can make an aerosol and you can, someone can inhale it and swallow it, right? Yeah.
Right. Right.
There was an
MMWR report of vacuum cleaner spreading norovirus after somebody had puked on the carpet.
My goodness.
Oh.
Really? Oh my
That's
gosh. So
it was at a wedding venue where there had been a vomiting incident, and then somebody at a subsequent party picked up the same strain of norovirus, and they traced it to the spot on the carpet where the vomiting incident
clearly did not have a Dyson.
Anyway, the idea is these things are not mutually exclusive.
Yep.
And it's really fascinating to read.
Cool.
Neat figure. Man, what a drop in miasma, huh? Holy cow.
Yeah.
Rich, what do you have for us?
So, I don't know if somebody might have picked this before in my absence. No? Okay. There was a little spree in the newspapers a couple of weeks ago about a digital reconstruction of the wreck of the Titanic, which caught my attention because they had just little video clips of the thing. And then I went looking around because I figured there ought to be some sort of interactive site or something where I can tour the Titanic. And if there is, I couldn't find it. But I did, in my touring around, find this particular report from BBC. Looks like it's on some sort of morning talk show that describes this thing. And there's a company called Atlantic that has done digital mapping of the Titanic. The images that we have of the Titanic wreck are photographic images that are limited and they're hard to see because it's murky down there.
The visibility in the North Atlantic is terrible.
This is what it's like several miles down.
12,500 feet deep.
Yeah. So that's what, 2.5 miles, something like that.
Yeah.
For mile people like me. At any rate, so they have, using various technologies that I don't understand, managed to do essentially a digital scan of the whole wreck. And this, I presume, ultimately, I hope, will be available in an interactive fashion once it's all assembled so that I can tour the whole ship and zoom in and zoom out and get an absolutely crystal clear impression of the whole thing. They do have a couple of clips, and in this BBC show, they do a trick where they put the reconstructed wreck on a football pitch that's English for soccer field.
Soccer field.
Okay. And it takes up the whole thing, right? And then they show some of the little clips. And I mean, it's detailed, sufficiently detailed so that you can see, trinkets, shoes, and toys that were left behind or scattered around the wreck. You can see a serial number on one of the propellers and that kind of stuff. So it's pretty remarkable.
It's neat.
It's cool.
It's very cool. And this technology, this mapping technology, has tremendous potential for mapping reefs and other wrecks. I mean, there's a whole history down there.
This has nothing to do with this, but ChatGPT, as you know, is being extensively used in all things. And over on TWiV, where we have clinical cases that people have to solve, now everybody's using ChatGPT to solve it.
Oh, I heard that in the last episode, and they even said I use ChatGPT.
They send us the transcript. It's like you're supposed to look stuff up, and it's the whole discovery process where you learn a lot instead of having someone hand you the answer. You're not going to learn anything, right? It's too bad.
I guess it would be interesting to do all your own research in this case, and then put it into ChatGPT and compare.
Yeah, you could
Maybe
compare
they could
it.
do
That'd
that.
be fine. But you can go and read some articles and make it.
Of course.
But gosh, it's people want the easy way out. It's interesting. Anyway, Alan, what do you have for us?
I have a site from Stanford University. They have this science education in an age of misinformation effort, I guess. And this is you can go to the About tab and see who's involved. It's some pretty big names in science. And they are talking about how we teach science and how we might reconsider some of the ways in which we teach science in the context of this sea of misinformation and disinformation that current students in schools are finding themselves in. Of course, the pandemic threw this all into stark relief, but it's not new. This has been trends that have been going on for a few years. And so the whole site has various articles about, they have a main report that they did on, this is based on U.S. science standards. Some analogies would apply worldwide, but the science standards that are used in U.S. schools are very much centered on teaching people how science works, which is a great idea in principle. But as they point out pretty persuasively here, maybe that's incomplete now, because in fact, and it may even have some aspects that are counterproductive. I had never thought about this before, but if you go to school and you learn, oh, okay, this is how science is done, so I am now qualified to evaluate science. Wait a second here. The problem, of course, is I'm not qualified to evaluate all science. Nobody is, not even a scientist, because if you are a microbiologist reading about climate change, well, it's not my field. How do I assess the quality of climate science? And they talk about assessing source credibility and all these basic rhetorical techniques that one would learn in something like a debate class, but that's not taught in the context of science education. So I found it very interesting. I thought it was good food for thought, and I highly recommend browsing through this for anybody, but especially, I know we have a lot of teachers who listen to the program. Anybody who's in education really ought to sit down with this report and think about it. And if you have any influence on how curricula are made, maybe consider some of these ideas.
This report looks pretty cool.
Mm-hmm.
It is.
That's good.
I love the picture that they have on the front page here of all these kids staring at their phones.
All these kids looking at their phones, yes.
Mm-hmm.
Yeah, and if you walk the streets of Manhattan, that's what people are doing. They're not looking around anymore. Even the home.
Oh, my favorite is seeing like 4 women walking across campus together on a wide sidewalk. and they're all looking at their phones. They're not talking to each other, but they're walking together.
And then none of them move out of the way when you're trying to get by because they're looking at their phone.
Right.
I hate that so much.
Yeah. I'm not going to go off the sidewalk. I just stop and stand still, and then they have to move around me. Yeah.
Well, the problem is when people are driving and they're on their phones, right? It scares the hell out of me.
There's that too. Yeah,
that's tough. All right, my pick is a article in eLife. Well, I don't know. It's not an article. It's a piece on what's happening. It's by John Dennehy.
A feature.
A feature.
That's what they call it.
Okay. Who's at Queen College and who's been on TWiV, and he's deaf, and this is all about how the pandemic was a real problem because going online was really hard. He has a cochlear implant, and he says that all the electronic, Zoom sounds, he couldn't hear any of it. It was all distorted because of his implant. And I remember when we had him on TWiV, he showed me how to turn on the closed captioning, which I'd never used before. So he had to explain it to me, but that takes everything we say and translates it into words on the screen, and then he could see what we're talking about. Otherwise, he couldn't participate. So he said when lockdown occurred here in New York, he was lost. He couldn't communicate with anyone, and he couldn't teach his classes. He had to get Google Meet because he said the software that the university used didn't work for him. So he got Google Meet to work, but he said his colleagues always had to use their own platforms, didn't have closed captioning, right? And he said conferences, forget about it. He couldn't understand any of it, and people said he was making life complicated for them by requesting, to get transcripts or real-time text and so forth. But
that's ridiculous.
It's totally ridiculous,
that's
right?
That's so ridiculous.
But he said within months, a lot of these online communication platforms started incorporating closed captioning into their software. And he said it changed, and eventually he was able to hear. And he said the problem is you have to ask for it. He said hearing loss is often an invisible disability, and hosts may not realize that attendees have hearing difficulties. They may assume that people will request the accommodations they need, but many are embarrassed to ask or don't want to be a bother. So it's better to provide them
because they don't want somebody saying you're making things complicated.
Yeah. Exactly. Yeah.
Well, and also, I can remember when we did that TWiV and watching the closed captioning and how it mangled what we were saying.
Yeah.
And so the deaf person also has to be able to interpolate,
sure,
or extrapolate, whichever it is, what they're seeing in the, in the closed caption into what it must really be.
Yeah.
And
especially scientific terminology, the closed captioning is so bad.
I do a lot of, a few years ago, I finally automated transcription services got good enough that I could use them.
Mm-hmm.
And I routinely now, when I interview somebody, I get an automated transcript of the interview. And it's great. But I would never expect somebody to understand what the hell was going on in the interview from these transcripts where all the scientific terms have been butchered.
Yeah.
And I can read through it because I did the interview and I know what was said, and I say, oh, okay. And then I can get the quote, and it's got timestamps so I can go back to the tape and get the actual transcript corrected. But trying to piece out a scientific conference by putting words together from that quality of a transcript would be a huge task.
I'm sure that there must be like words that he's now, him and other that have any sort of hearing impairment, that they know that it's captioned in a certain way, and but then they have it's twice as difficult because then they're like, oh yeah, this means this and this means this and this.
That means,
he's got a wonderful paragraph in here where he describes his hearing impairment.
Yeah.
And what it's like. Very, very interesting.
Yep.
For me, hearing isn't the problem. The problem is making sense of the sounds. Picture yourself in a foreign country where you have only rudimentary understanding of the language. Okay. So there's only a fraction of what's being spoken that comes through, and he spends a lot of effort trying to figure out what's going on. Amazing. And it looks like the bottom line here, I've only been able to skim it here, Vincent, is that once these platforms caught on, things got like a whole lot better, right?
Yeah, for sure. And you said it. The whole environment's now changed because of the pandemic, and that all of this is easier, which is good. But it just took time.
Yeah.
I mean, the YouTube that's going to show this podcast, it does live transcript. You can see the words scrolling through on the right. It's not bad. It doesn't.
Yeah, the having just bagged on automated transcription, they have improved drastically in just the past year and 2 years, really. So I think the pandemic definitely drove improvement in that technology.
So it's a cool article because I remember he would watch. I could see him watching the transcript, and you had to wait because we all just talk really quickly and don't wait much, but for him, you had to wait because then it would take him a while to react, and you could tell he wanted to say something, and often he got cut off. But he has to be aggressive and just push his way in there. Otherwise, he's never going to get anything said. But he did. He participated very well.
Yeah, it was great
in the discussion. So, all right, that is TWiV 1013. You can find the show notes at microbe.tv/TWiV. You can send your questions, your comments, your picks to twiv@microbe.tv. If you enjoy what we're doing here, we'd love to have your support. Microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Thank you. Always a good time. It's great to see everybody.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
And Angela Mingarelli is at McGill University. Thanks, Angela.
Thank you all. I always love being here, and stay cool. It's very warm. I mean, we're all 90 degrees almost.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.